So it's my pleasure to welcome to the Language and Brain seminar, Dr. Lorna Quandt. So I'm honored to have the privilege to introduce Dr. Quandt to you all, who I've known and been friends with for almost a decade, and who I consider one of my role models as a researcher and as an overall human being. Dr. Quandt received her bachelor's degree in psychology from Haverford College, then pursued her PhD in cognitive neuroscience at Temple University, where I met her as we were both getting our PhDs. At Temple, Lorna's research focused on the neural correlates of action and action observation using EEG. As a postdoc at the University of Pennsylvania, where we also overlapped, Dr. Quandt examined the intersection of action representation and language. Using fMRI, she contributed novel insights into how abstract action words might be different in the human brain from actual movements. Since joining the faculty at Gallaudet, Dr. Quandt has woven these research threads together. Her lab now examines the sensory motor contributions to meaning in sign language. Combining EEG and behavioral measurements, and very recently supported by a grant from the NSF, she's also been incorporating virtual reality, something I'm particularly excited to hear about today. On a personal note, as I mentioned, Dr. Quandt is a close friend and role model. As evidence of this, the astute among you may have noticed that Lorna and I both went from PhDs at Temple to postdocs at Penn. One might say that I sort of followed her to Anjan Chatterjee's lab at Penn, joining 1 year after she did. Of course, I partially joined Anjan's lab to work with Dr. Chatterjee myself, but frankly, I would join any lab that would have Dr. Quandt as a member. So join me in welcoming Dr. Lorna Quandt. I hope you're as excited as I am to hear her talk, which is entitled Sign Language Expertise and Embodied Cognition, drawing from EEG, behavior, and emerging technology.
Wow, thank you for that introduction. I realized today when I was thinking about this talk that it had indeed been about 10 years since I first met Steve, and that made me almost tear up a little bit, but I won't reflect too much on that because that's not what you're all here for today. But thank you really for the invitation. Happy to see some friendly faces in this audience, and thank you all for coming today. Okay, I'm going to share my screen in Keynote, so I will have to check with you 1 or 2 times to make sure that you're seeing what I want you to be seeing. Okay, so share screen, Keynote, share. Are you seeing what I want you to be seeing? Do you think?
That works.
We see a title slide.
Okay, that sounds good to me. Okay. All right, I've lost your faces. Okay, one moment, let me get situated. So Steve told you the title of my talk, and what I will be going over today is, as he mentioned, some of the work that I have been undertaking during my time at Gallaudet, where I started my position about 5 years ago. And wow, that time has really flown. It's hard to believe it's been that long, but also it's gone by just so quickly. Okay, I make sure I can see you all. Hold on, it's disorienting here for me. Here we go. Okay, much better. It's hard to give a talk if I can't see anyone's faces, but I got it. Can you guys see the next screen? I just advanced slides. Okay, perfect. We're in business. Thank you. Every time I give a talk on Zoom, I need to take that 1 minute, but we are in business and ready to go. So first I'm going to Steve set me up really well. So I think he gave you an idea of the background of how did I get to this point? Why am I studying sign language? So For much of my academic career, I was focused on actions as actions, things we do with our bodies and our hands specifically. And I had great fortune in my career in being able to sort of look at the intersection of actions, particularly hand actions and gestures, overlapping with language. And at an exactly right moment in my career, I came across a job posting at Gallaudet University, and the opportunity arose to study sign language, which is, of course, at the intersection of hand movements, gestures, language, and we can see here why we would be studying sign language through this lens of action, communication, gesture, and meaning. Can you see the video moving on my screen? If you are familiar with sign language, you already know this, but if you don't have a lot of familiarity with sign language, here we see students communicating in American Sign Language. ASL is extremely complex, three-dimensional, utilizing the space in front of the signer in a systematic way. They are placing concepts in space, referring back to them, using time and space in a structured manner. You can see that they're using their bodies, their hands, their facial expressions in a highly systematic way. And I felt like the more that I learned about the structure and the way that the body and language intersected in ASL, the more I realized that there were hundreds of questions that needed to be answered about what is going on with people who grow up using American Sign Language. Of course, there are many researchers who have been working in this field, but I feel like the research questions are still untapped. There's so much there that is yet to be figured out. So I'm going to tell you a little bit about what I've done. I've only begun scratching the surface, of course, but there has been a lot to ask. So my interest was piqued because it's clear that sign languages, I study American Sign Language because I live here, but there are hundreds of sign languages that are used across the world. Sign languages are embodied, they happen, they occur on the human body. They are embodied, spatially complex, and three-dimensional. So how does the experience of growing up using a sign language, or maybe the experience of learning a sign language, how does that experience change other things, like how you see the world or how you see other people's movements? So those are the kinds of questions that I wanted to start investigating. And I knew from my own prior research in other related domains, we already knew that action experience in general can change perception, but we didn't know much about how sign language experience can change perception. So I'll walk you through two different studies that I looked at through this sort of lens of how does sign language experience change perception. Before I go into those two studies, I will say that because I'm trying to go through two different studies, we won't be able to dig really, really deep into either one of them. I'm happy to answer any questions that you have while I show you the data. And certainly if you want to stay for more questions at the end, or if you have time to stay for chatting at the Q&A, or I forget what Steve called it at the end, but social time at the end, I'm happy to chat more. Also, I'm easily available if anyone is desperately curious. I can always be reached by email, or you can hunt me down and ask me real questions anytime you want. So I also want to put out there that I'm going to frame this a little bit like about ASL experience, but the most common question I get about this work is like, is it ASL experience or is it deafness itself? So before you ask that question, I will say I'm not sure yet, because in order to answer that question, I know what we would need to do, kind of, but it would be incredibly complicated and no one has yet given me all of the money to do it. So I need a lot of money and a lot of people and a large team of researchers. So the research I've done so far has been comparing people who already know American Sign Language fluently and people who don't know American Sign Language. And the people who do know ASL in my studies so far are deaf people. So we're comparing deaf signers with hearing non-signers. So it's either a confound or not a confound, depends how you view it. In order to get around that sort of problem, we would need to also study hearing people who grow up using sign language and also deaf people who don't know sign language, or we can also bring people into the lab and teach them sign language and study them as they learn sign language. But we haven't done all of that work yet. So we'll figure it out one day, but it's very complicated. Happy to hear your ideas about how we can figure out that question, but it is something that I'm well aware of not being able to answer yet. So first I wanted to look at this question of if ASL experience changes the neural substrates of sign imitation. So this is a pretty basic question. If we put two groups of people, deaf ASL signers and hearing people who don't know anything about sign language, if we put them in an experimental paradigm where they see a sign and then they copy that sign, what is going on in their brain while they are doing that task? Particularly, I wanted to focus on sensory motor EEG rhythms. So it was an ASL imitation task, see a sign, copy a sign. And we had 18 hearing participants and 19 deaf signers. And they either saw one-handed signs or two-handed signs. And this is important because if we view the signs more through the lens of like an action, one-handed dominant hand action would primarily use the left sensory motor cortex, whereas a two-handed action would use both sides of the sensory motor cortex. If you're deaf, this task is like a word imitation task, pretty rote, pretty straightforward, pretty easy. If you're hearing, it's a more complicated task that actually ended up causing a fair amount of stress for our participants because copying novel gestures after one viewing is very stressful for a lot of people. So I'm going to show you the EEG time frequency results of what this looked like. So this is while they're just viewing a sign with the intent to imitate. These are EEG time frequency results. I'm going to check with you all. If I move my mouse on my screen, can you see my mouse? Okay, that helps a lot. Okay, so time 0 is where the dotted line is, and that time 0 is at the onset of the sign. So this is the video that they're seeing. Time 0 is when that sign it sort of starts. There's a way that that's defined in the literature that we used. And you can see that for deaf signers, there's a difference in one-handed and two-handed signs. This is alpha and beta responses at a centroparietal electrode over the sensory motor cortex. And we see that two-handed signs, during observation of two-handed signs, they are eliciting somewhat more sensory motor response, suggesting that maybe they're using some more of their sensory motor cortex in something like a mirroring-like response. They are using more of their own sensory motor cortex to process the sign that they're viewing. That would be one interpretation. For these deaf signers, that process is starting early. It's even starting right before the onset of the sign as the hands are coming up to produce that two-handed sign. And you can see that significance in the third panel. That's the significant difference between the one and the two-handed signs. Whereas for hearing non-signers, there is a significant difference between the one and two-handed signers, but it's coming later, at least at this one electrode. This type of pattern was evident at many of the electrodes across the general central region over the sensory motor cortex. So this supported our prediction that for deaf signers, there would be earlier and more robust differentiation between the one and two-handed signs. Although, to be honest, this was one of our predictions. We were not 100% sure which way this would go because there was also reason to believe that deaf signers would be less likely to be processing these as actions and would be more processing them along a linguistic only, not really taking in the sensory motor details and attuning to the characteristics of these motions. I'm happy to go into that more in the Q&A if anyone is curious about these ideas. One thing that I will emphasize is that the intention to imitate was really important here, because we did a similar study without the imitation component, and we did not find the same pattern of results. So the fact that our participants were all about to imitate this, the signs that they saw was very critical for this pattern of results. So from this finding, we did conclude that we saw an earlier and more robust differentiation in deaf signers compared to hearing non-signers. One-handed and two-handed signs. And this same pattern, this is just another way of visualizing the same type of data across the scalp. So you can see it wasn't just at that one electrode. But again, we have deaf and hearing participants. And again, these one-handed and two-handed signs. So here you can see that this effect was significant at different clusters of sensory motor, sort of central sensory motor electrodes at a variety of different time points. Okay, are there any questions people want to clarify about these results at this time? I'm happy to take a question break if people want. Okay. All right, so again, from this study, we did conclude that we had earlier and more robust differentiation in deaf signers during this imitation task. Okay, so we conclude that ASL experience or deafness changes the neural substrates of sign imitation. And again, emphasizing that, of course, the task was really different for these two groups of people. So it's not that I was surprised that their brains would be doing different things, but this work had not been done yet. So we didn't know what kind of differences we would expect there. So the next study I'll show you is looking a little bit further outside of what kinds of effects we might expect to transfer. So how about signers seeing other kinds of movements, non-sign movements? We asked here whether ASL experience impacts biological motion perception. So in that first video, one thing that I tried to emphasize with that video is the way in which, if you are experienced and fluent with sign language, it doesn't really matter if you're standing right in front of another person signing. You can still understand them just fine if you're sort of to the side, you're seeing them from a non-canonical, not perfect perspective, it's fine. Your brain is pretty good at picking up what they're saying. Just like if you're speaking and you're a native speaker, you can hear someone just fine, even if it's over the phone and it doesn't sound great. Your brain can put it together. So what we wanted to do was to see whether native ASL users who grew up with ASL as their first language would show a different or enhanced ability to extract biological motion information, even when it was presented through point light movements and from rotated perspectives. So quite degraded. So we have a motion capture studio at Gallaudet University, and we created point light displays, both which were pretty easy to see, like that jumping jacks that hopefully you just saw, which moves straight on, so it's pretty easy. But also we recorded them from a oblique angle, and we had 3 different angles. So some of them were more difficult to parse. So this one is a little more tricky. So we had 6 different actions, and each of them had 3 different viewpoints. So we had 18 different stimuli, which we kind of wanted them to be exactly like in an organized way, to be, an easy, medium, and hard of each one, but that did not pan out because it turns out it was hard to predict which would be easy, medium, and hard. But there was a variety of difficulties, and well, I'll show you the data, but basically we had this nice stimulus set with a variety of everyday actions. And first we asked a lot of people using an online survey, we asked them just to self-report first what they saw, and second, how hard was it to identify what you saw. And from that, we had a pretty large sample size of hearing people who didn't know anything about sign language, and a smaller sample of deaf people who do know sign language. And just from self-report, we see that deaf signers self-report much easier identification of these actions than hearing people. So take that as you will. This survey was not linked to any other surveys. They weren't being asked anything else about language or any other information about the actions. So it wasn't tied to any other sort of like leading questions, but the deaf group self-reported much easier identification of these point light display activities compared to our hearing folks, which ties in nicely with what we found with the EEG study that we followed up with. So for the EEG study, which was with an entirely different sample, none of the same people, we had a very passive task where we showed people these point light displays, and we tried to come up with a very passive task just to have them attending to the stimuli. So we asked them to just tell us after each block how many of the stimuli you saw involved a ball. So that they're just kind of trying to identify them, but we didn't want them pressing buttons so that they would, not to get any sensory motor interference. So how many involve a ball? Give us a count every once in a while. And from that, again, our original intention is to look for sensory motor primarily over central region electrodes, to look for classic sensory motor involvement in alpha and beta sensory motor rhythms in the EEG signal. But we also performed full scalp analyses to look for possible occipital and frontal effects. There has never been EEG studies of point light perception in deaf signers before, to my knowledge. So we did have to kind of shine a flashlight on this to set a foundation of what will we find and what is going on here. So we put forth our predictions while also looking at everything so we can, figure out where to go next from here. So what I'll show you today, we have time frequency results. Here is electrode FC5, which is a frontocentral electrode on the left hemisphere, right around here. And I'll use my pointer here. So on the top panel, we have deaf native signers. And what you're looking at is a time frequency response from starting at 0 milliseconds, and that's the onset of the point light display stimuli. And those are exactly like what I showed you guys originally with the jumping jacks and the like oblique kicking. So it was the onset of that kind of video. And those videos were 2 seconds long. So that's the time on the x-axis. And on the y-axis is the frequency. So starting at 4, we included the theta band, going up through alpha, starting around here at 8-ish, and going up through the beta frequency up through here around 24. And that these scales are the same for all 6 of these panels. So we had biological motions, which are what you saw. And then I didn't clarify when I mentioned the stimuli that we also had scrambled controls. So the scrambled controls were made in parallel to be a control group to the biological point light displays. The scrambled controls were the same dots moving the same way, but the locations were scrambled. So they moved, but they didn't move to show you anything recognizable. They moved just to look like a jumbled mess. So we had that scrambled control and the biological motions. And on the top panel, we have the deaf signers and the significant difference between those 2 conditions here. And on the bottom panel, we have the hearing non-signers and the significant differences here on the bottom right. So here we have our differences between the 2 groups, and this is pretty characteristic of what we saw over most of the sensory motor region is earlier and more consistent differences among the deaf signers, particularly on the alpha and beta EEG ranges. A later response or less strong responses in hearing non-signers. So we concluded from there that we did have enhanced and earlier differentiation in deaf signers. And I can show you, I think my next picture is where in the brain these different, or where on our electrode map these differences occurred. So we did have a lot of frontally located differences. So a lot of this is frontal with some central differences occurring later. And like I said, a lot of what we have here is the first time that we have ever analyzed data from this type of stimuli in this type of group with this type of analysis. So while we did have our predictions going in, we also do feel a little bit like, you know, it's a very rich data set with a lot going on. So we are submitting it for publication soon, but we don't have all the answers yet. So we're already like planning follow-up studies. We're welcome. We're ready to hear anyone's feedback or ideas about these data, because it does feel like we, you know, have a lot and we have a lot of questions going on here. Any questions that I can clarify for anyone about these data or the last slide that I showed?
Lorna, I have a quick question, just because I'm not accustomed to seeing this kind of data. And if you go to your previous slide, you know, you get this sudden loss of activation in the alpha region. And I just wondered if you could like talk to us a little bit, what's happening there at 500 milliseconds that like, boom, you know, is that pretty much standard anytime you show some kind of visual stimulation that there's this like quick response and then at 500 milliseconds? And it's particularly interesting because the non-signers, all of their significant differences are late, you know, after you get this sudden like poof, no more alpha. So could you just explain a little bit for us? Thanks.
For sure. I know. So sometimes when I present time frequency responses, it is like a lot visually to show. So I totally understand that there are many dimensions of information going on here. So for point light display EEG experiments, it is common to see around like 300 to 500 milliseconds, a onset of, I would call this alpha or beta desynchronization. So a abrupt reduction in power of that frequency. So it means that compared to the baseline we used, which the baseline is right before the point light display video starts, so just prior to time 0 was our baseline. So compared to that baseline, about 500 or 300 milliseconds following the onset of the video, we have less alpha or beta power present. So just that amount of power decreases in the EEG signal. So that could be for any number of reasons, in particular, depending on where in the brain you're observing that pattern. So in frontal regions, that might be because you are exerting greater cognitive control for some reason. It could be because you're accessing memories that allow you to make sense of the action you're seeing. It could be because you're integrating what you're seeing with a memory that you have from your own life that allows you to label the action you're seeing. If it's over a sensory motor electrode, it could be because you're activating your own sensory motor program for that action yourself. If you saw that pattern over occipital regions, it could be a basic visual process that's being brought online. So it does really matter where in the brain the pattern is being seen. So it is important to view it as like a time-dependent activity that is being shown. This, like the blue blob is just like a change in level over time. It's not an absolute thing that's happening at that moment. Hopefully that helps. So yes, around 300 milliseconds at electrode FC5. What I think is happening at this time is that that more like sensory motor processing is kicking in, that you are able to make sense of and sort of use your sensory motor network to process the differentiation. Here, what you're looking at is scrambled versus biological. So you are processing the difference between, oh, that's a real movement. Oh, that's someone doing a high kick, compared to that's a scrambled bunch of dots that doesn't mean anything. So it appears that that is happening earlier and more successfully for these deaf native signers. Great question. Okay, any other thoughts on these ones?
Yeah, we have another question by Leah. Leah, we'll type your question in the chat. Hold on.
When the question is typed, will someone read it? Because with however Zoom is right now, I can't like see anything.
Yeah, I can do so. Okay, so Leah's asking, were the control group watching the scrambled movements primed to interpret them as biological movements, or did they just know they were seeing random dots?
Great question. So all of the participants were given the same task for this experiment, and all of the participants were watching a randomized order of these dots moving. And so every time a new trial started, the dots would move. And if it was scrambled dots, then you just see the dots move. And after a moment, you would know that they were just randomly scrambled dots moving. And if it was a biological coherent movement, you would see like, oh, that's a basketball throw. So you would mentally think, okay, 1, like that's a movement that requires a ball. And then the next trial would start and it would be a jumping jack. And you would think, okay, oh, that's one that doesn't need a ball. Okay, I don't need to count that one. Then the next one would be scrambled. And so you would not count that one. So they were randomly scrambled or non-scrambled. And so if it was a biological meaningful movement, then you would have to attend further to make sure you knew whether a ball was involved in that activity or not. I hope that answered your question. So there were a variety of tasks that we considered. Other similar experiments have used things like attending to count how many trials had like a colored dot appear on the screen, things like that. But we wanted to think of a task that really required you to think about the type of action being done, and something like the color of a dot on the screen we thought was too much of like a surface level attention. So we wanted you to have to really identify what is the movement, not something like about the basic appearance on the screen. So I'm pretty happy with this task that we came up with, but we'll see if someone comes up with a scathing criticism of it. You know, let me know if you have one. Okay, I'm going to move on to this next part. Okay, so we do think that native ASL users do show enhanced differentiation between biological and scrambled motions, but there is a lot more work that we want to do to parse this further because, like I said, this is the first time we've seen something like this, so there's a lot more to do. And the last part I'll show you here is really a more sort of applied and translational aspect of our work. So this is a project that we received an NSF grant for maybe almost 2 years ago. And it has been a collaboration between me and my colleague, Melissa Malskoon. And this is a project in which we are taking what we know about human learning and sign language and trying to do what we can to hopefully build a better way to learn American Sign Language compared with some of the current ways that people learn sign language. So I'm going to show you more about that. And the goal of this is not just doing something because it's cool, but really trying to capitalize on some interesting emerging technologies to build a better tool for learning ASL. So I will clarify, I don't mean better than like an in-person class with a skilled teacher, because there's probably nothing better than that. But there are some amazing technologies out there in the world that have not yet really been applied to teaching and learning American Sign Language yet. And we think that we're ready to make that happen. And so we've been working for the past 2 years to try and make that a reality and kind of build a proof of concept for that. Specifically, the project is called Signing Avatars and Immersive Learning. So I mentioned before that we have a motion capture studio in one of the labs that I work in. So we use that motion capture studio to build amazing, I think, amazing signing avatars who can really sign with the qualities of natural human signers. So their movements are not like the movements of a computer synthesized signer. So they're not moving like a robot or like something built by a computer program, but they're moving like a human. The downside is that that's very demanding and requires a lot of human labor. But we put those avatars into a three-dimensional world that we access in virtual reality. And the benefit of that is that because, as we previously discussed, American Sign Language is a three-dimensional space, or is a three-dimensional language, we capitalize on the richness of that three-dimensional nature of the language and the three-dimensional nature of immersive virtual reality. And we are starting to create a learning experience that can create something richer than, for example, learning ASL using YouTube videos or other things that people do when they don't have access to high quality in-person instruction. So I do have a little video that we made for an NSF competition. So I think I'll show that video. Since we put a lot of time into the video, it makes more sense to show you that than me spend 3 minutes trying to recreate it as best I can. I think the best thing for me to do is like stop sharing Keynote and then start sharing that. Does that work, Steve? Okay. Okay, stop share and then I'll reshare. Okay, so I will share in just 1 second once I have that back. So this project, here we go. Okay, can everyone see my Google Chrome with a video about to play? Okay, great. So yeah, this video introduces the project, and this project has been underway for a couple years, and we're hoping to extend it with a new sort of bigger and better version, hopefully in the next couple of years. And we do have a better version of the project than what you'll see in this video, but it does give you a pretty good idea of where things are. Let's see. Can everyone see enough? Okay. So I am, let me see. I'm pretty much wrapped up with everything except for my thank you and questions slide. So I did come back to that because it's important to thank all the people I've worked with. And I have a great team at Gallaudet and amazing collaborators and students and research assistants and everyone there. So it's been a great time. I have a great team of colleagues, and I'm very, very thankful that I did come into this field, and I'm thankful for the invitation to share this work with you all today. So happy to take any questions that you all have, and I'm happy to stay and chat with everyone as much as you all will have me. So it's been a pleasure so far.
Thank you so much, Lorna. I think people can just unmute themselves or put the questions in the chat, and I'll also stop recording just so people are more free to ask the questions.